So Vito says he is on the link, but did you see whether he was, it doesn't look like anyone new joined. I'm hoping it's not a different link that we've got.
I see
So
two
I just
lists.
added
I see
Vito, so
panelists
he
and
should be able-
attendees. He's an attendee.
Oh, there we go. Yep. All right. Now the attendees are showing. Perfect.
Okay.
I don't see anything. Hi, Ben.
Hi, Vito. Thank you so much for the invitation.
Well, you have a double invitation now. I didn't
I know.
realize there was, yeah. So,
But
but this is special.
I'll try not to give the same. I'll try not to give the same talk in 2 months. That was the goal I talked about with Christine a little bit.
Yeah. So yeah, we just didn't communicate, but
It's okay.
this is really, was it explained to you? This is like, so students are involved in this, and do we have you booked for an hour after this also?
Yes.
Okay, good. You met Kavya, Sherman, there, of course, very tight voice, okay. Do we have attendees at all? This is the first of the fall, and so I don't know whether anybody's going to show up. Hopefully they will.
It looks like there's 13 so far. I guess
Okay,
Darren, Lewis, and Kavya, if you have questions, I probably won't see yours because I have to click between either the panelist list or the attendees list. So I'm just going to stay on the attendees list.
Okay.
That sounds great.
I don't see anything here. I don't have access to anything.
Hold on, Vito, let me see if I can add you as a
We
panelist
purposely
real quick.
excluded you.
It's okay. I mean, I don't need to, you know, if I don't know, it's better. We usually for this systems biology series, we have about, 25 to 30 people. So it's not a big major seminar.
Mm-hmm.
The other one that you will give will be a bit bigger, hopefully. But this is usually 20 to 25 people. And I'm not sure if they're still, this is the first of the semester. I don't know if people are into it yet.
No
But,
worries. I didn't attempt to fill up the hour with 70 slides, so we're good.
Right. But so it's good also that you and then you have a repeat in case this is a little light. So you're in Dallas, right?
Mm-hmm.
Nice.
I am. I moved home.
Oh, so you're original. So everybody from Texas eventually
Yeah.
goes back to Texas.
Yeah, I grew up here. I just I left for college, but so we just moved back February.
Oh, nice. And what department are you in? Medicine or something else?
Yeah. I'm in internal medicine. I'm in the hemonc division.
Okay.
Because I just started seeing patients. They gave me a little bit of a grace period to get the lab set up, and then a little bit more of a grace period because it's not a normal year.
You might have heard there's a guy there, John Minna, you may want to talk to.
He's about 100 feet away, and he is the reason that I'm here. He called and invited me back, so that was it.
Yeah, you couldn't say no, of course, right? Texas is calling back.
There was no reason to say no.
Right. So, no, but you see, Southwestern is a fantastic place. So I visited there maybe 2 years ago. Yeah, I came there. There was a group. Now they left, but there was a group in systems biology, computational biology, actually.
Mm-hmm.
But they moved. Let me see, what department were they in? Is that guy Rana? I don't know if you have heard of him. Rana. What's his name? He's a structural biologist.
It is a massive place. I'm still.
I know. But this guy Rana is a special guy. I'll send you his name. He is really worth talking to. He's one of these. I think he's an MD PhD also, but really got deep into structure.
Cool.
So,
all right, Vito. Why don't we get started? We've got 15 attendees, plus the 4 of us on here. So.
Okay, so let me go back to this. Okay, so shall I start?
Yeah, please.
I'm gonna talk for a few. Okay, so everybody, welcome to the fall series of seminars for the Quantitative Systems Biology Center. We have a special guest, today, and also a special time. These seminars, as you will see, will occur on Wednesdays. And before I go any further, I'd like to thank the people that made this possible. And so, first of all, Lois Austinine, and she is the one that sends you emails and sort of keeps these things and actually figured out how to make webinars work on Zoom. Kavya Sherman, who used to be, well, she's still a student, but she's the student that initiated actually these conferences, knocked at my door with Cody Iser and said, "We gotta do this," and I couldn't say no. And actually, Kavya, this year, she will work as a teaching assistant for the second part of these seminars. It's a 1 hour discussion with the speaker, which is actually part of the course CPB 8328. It's actually in the book, in the Vanderbilt catalog, Systems Thinking in Biomedicine. And that's a one credit, course for students that may be listening, that looking for that elusive, last credit, for the semester. And the instructor for this will be Darren Tyson, who's here, and he's the one that is you're going to hear from him mostly. And here he is. He's the one who's going to introduce the speaker for today. Darren.
Yes, thank you, Vito. Yeah, it's an honor to be taking over this course from you. It's one of the courses I think the students can get the most out of in terms of conceptual thinking around systems biology, their introduction to it, and the ability to think in terms of these interacting parts of a very complex system. So I have the distinct fortune of being able to introduce our first speaker, the inaugural speaker for the year, Benjamin Drapkin. And you've heard a bit about him already from Vito as a new assistant professor at UT Southwestern, as a new hire as part of their Cancer Prevention and Research Institute of Texas. So it's a very distinguished position that was offered in a nice startup package too, I must say. It was a nice draw. So, and they were lucky to get Ben there to join UT Southwestern, but he has a fantastic pedigree. He started his undergraduate degree at Yale and did his MD PhD at Mass General and Dana-Farber Cancer Institute, and his fellowship there. He's got a number of awards from, including the Young Investigator Award for the American Society of Clinical Oncologists, and has got research funding from a number of big pharma and the Lung Cancer Research Foundation, so Novartis, AstraZeneca, Merck, AbbVie. A number of big time pharmaceutical industry is looking to him as an amazing resource of knowledge in the field. So we are very fortunate to have him as our first speaker. Thank you so much.
Thank you very much. Thank you for the kind introduction. Thank you for the invitation. I'm gonna go ahead and share my slides and let me know if this is this working?
That looks great.
Yes.
Cool. Wonderful.
So just 1 second, Ben. If anybody has questions as we're kind of moving, Ben will kind of pause throughout the talk. So feel free to raise your hand. There's a button at the bottom of the screen for you to do that, or you can drop something in the chat or ask a question and that can be answered live. So feel free to do that throughout the talk. Thanks.
Okay. All right. So I'm a brand new hire at UT Southwestern. I just moved here from Boston to Dallas. And today I'm going to talk about my new lab's research efforts and where those came from during my postdoc. to study small cell lung cancer and a specific aspect of small cell lung cancer, which is resistance to chemotherapy and DNA damaging agents. So a little bit about small cell first. So small cell is a very common malignancy, about 33,000 cases a year. Far fewer cases than prostate cancer, but about the same number of patients die of this disease because the prognosis is so poor. So the average median survival is about 11 months, and this hasn't really changed very much for the last 30 or 40 years. And it's unique among solid tumors in that it's essentially non-surgical. So a lot of the time for breast cancer, other lung cancers, we will attempt to cure the disease by surgically removing it. And in small cell, this is almost never possible because it disseminates so early. There's also no targeted therapy for small cell lung cancer, so there's really no reason to resample the tumor after the initial diagnosis. There's no biomarker that we would be looking for in the patient's tumor. So all of this sounds pretty grim, but remarkably, it's actually one of the most sensitive tumors to therapy initially. So we measure sensitivity to therapy, the biologic response to the tumor, by something called a response rate, which is how much does the tumor shrink. If it shrinks by half, 30% reduction in diameter, then we call that a clinical response. And 70 to 80% of patients with small cell lung cancer have their tumors shrink significantly and often dramatically with first-line therapy, which is a combination of cisplatin cross-linking, DNA cross-linking agent, and etoposide, a topoisomerase inhibitor, so all targeted at DNA damage. And the problem with small cell that's bedeviled the field for 40 years is that it always relapses, kind of no matter how much chemotherapy you give or other DNA damaging therapies you give to the patients. And when it relapses, instead of, with the second line therapies, instead of a 70% response rate, now you get on average a 10 to 15% response rate. So it's acquired some significant resistance. So this is the main clinical problem for small cell, which is cross resistance to, we would call, when it becomes resistant, that it becomes resistant not just to the therapy the patient received, but also to a broad range of therapies. The term would be cross resistance. And that's the challenge that I'm trying to study. And so what do we need in order to study clinical cross resistance? The model system requirements would be that you need enough different examples of different patients' tumors to model the disease effectively. So a population that captures intertumoral heterogeneity. Those models have to recapitulate what the human disease looks like molecularly at a genomic and expression level. And probably most importantly, they have to recapitulate this part. I'm not sure if y'all can see my cursor. But the clinical responses to first and second line therapies. So one of the problems with the established cell lines is that they didn't do that last part particularly well. So in my postdoc, I set off to make these things called patient-derived xenografts, which are generated by implantation of a patient's tumor directly into an immunocompromised mouse without trying to grow a cell line or having it grow in vitro first. Most of the time, historically, for the last 15 years or so, people have been doing this. They do it from biopsies or from surgical resections. But as I mentioned, can't really do that in small cell because there aren't very many of those. So instead, during my postdoc, I isolated circulating tumor cells. So these are malignant cells shed by tumors into the blood that are probably the precursors of metastases, at least by hematogenous spread. Grow these, so isolate them, purify them in an unmanipulated way so that they're alive, put them into an immunocompromised NSG mouse, and grow the xenograft, and then start doing things to compare those xenografts to the primary tumors to see if they match up. The apparatus that I used for the last 5 years was called a CTC iChip, which functions by separating by size and then focusing the nucleated cells into a single file so that the leukocytes can be deflected, and so it's a process of negative selection to get the circulating tumor cells. And the main advantage of going with CTCs is that you can resample. So putting a needle into a patient needlessly or a scalpel, we really try to avoid doing that, but we can get a blood draw for research purposes, and this gives us the opportunity to derive serial models from patients. So over those 5 years, from 2015 to 2019, I derived 72 new models of small cell lung cancer from 50 patients, and the library that I came to UT Southwestern with is shown here, where each model is represented by a circle, red circles for CTC derived, blue circles for biopsy derived. Sometimes we derive two at the same time. And then the arrows, which are not drawn to scale for how well a patient did or didn't do on therapy, represent each line of therapy. The heavy black arrows representing the common first line therapy, which is platinum, carboplatin or cisplatin plus etoposide. And because I was deriving these models largely from circulating tumor cells, about 2/3 of them were derived post-relapse at that time point when the disease actually becomes lethal. Because it's upfront, it's very sensitive to chemotherapy. And so I will briefly go through, I'll just describe here the first comparison that we did of these models to the primary patient tumors to assess their molecular fidelity. And this was a genomic study comparing whole exome sequencing of the patient's biopsy to PDXs that were derived either at immediately at time 0, so the first tumor to emerge, or at passage 2. And I'm summarizing here iconographically. We did this for 7 different models. 5 of them were derived from CTCs and 5 of them from biopsies, and there was a greater than 95% identity of the molecular alterations for all 7 of these. And so for biopsies, it's good. It means that there's not a lot of clonal selection in the mouse, but we're in most of the time for those biopsies, we're comparing it to the biopsy from which the model was initiated. So the same tumor. The CTCs, this was a little bit more surprising because there we're comparing models derived from cells circulating in the blood with a biopsy that's at an anatomically distinct location, and yet it's still, there was greater than 95% identity. I think this speaks to what seems to be a hallmark of small cell, which is that although it has a very, very high mutational burden because it almost always arises in smokers, most of those are passenger mutations, and actually it's fairly clonal. And this is what we've seen in the large genomic studies of this disease as well, as opposed to some other tumors that might be marked by a high degree of subclonal heterogeneity at the genomic level. So the next couple of slides, I'm going to go through the functional fidelity of these models, but I'm not sure if, so it turns out I can't actually see the, I can see. So if anybody had any questions at this point, I'll just scroll through. I think we're good. So I'll go on with the comparison of how do the models respond versus how do the patients respond to standard first-line agents that we give for this disease. And the 2 tumor metrics that I'm going to describe, all of these experiments are in vivo in mice, the 2 tumor metrics are going to be response and time to progression. So response is going to be how much does the tumor shrink from when we started it. Time to progression is going to be how long does it take to grow to 2 times the initial volume. And for the first line therapy, I'm going to compare 13 models derived from platinum naive patients, so chemo naive patients, versus 19 models derived following relapse after at least 1 line of therapy. And what we found, so this is the sort of standard way in which we look at this in a clinical trial. It's called a waterfall plot, where the bars represent how much the tumor grew or shrank, its maximum shrinkage after the initial drugging window. So a positive number means it grew straight through. Negative number means that it shrunk at least somewhat, and the purple bars are from the post-relapse models. The green are from the naive models. And overall, unlike cell line models, this system is capturing the fact that the post-relapse models should in aggregate be more resistant to chemotherapy than the pretreatment models. And within this subset of relapse models, if the model came from a patient that had a long-term response to platinum and etoposide, this is this chemotherapy-free interval that should say greater than 90 days. In general, they tended to be more sensitive than models, post-relapse models from patients who relapsed quickly. So that's the first line therapy. The second line therapy that I'm going to show is called, it's a different combination of drugs, Olaparib, which is a PARP inhibitor, which will inhibit the cell's response to DNA damage, and Temozolomide, which is a DNA alkylating agent. And the reason I was studying this was because we were running a concurrent phase 1/2 clinical trial of the same combination in post-relapse patients. Shown here, this is actually in people. It was pretty effective for a second line therapy or relapse therapy in small cell, with an overall response rate of 42%. So 42% of the patients had significant shrinkage of the tumor. From the phase 1 portion of the study, from 4 of those patients, I was able to derive models, including 2 models, both before and after therapy for 2 of those patients. And so these are shown here. So on the left, you see the names of each patient, the MGH 15, 18, 28, 43. And the green arrow points to their, the tumor that we were following before treatment and then on treatment, where it shrank quite a bit for this patient that we're looking at here, and then 208 days later when it started to progress. And for this first patient, 1518, I was able to derive a model both before treatment and following relapse. This model before treatment was actually EP naive as well. And what's shown in the spider plots is how the mouse model responds to Olaparib temozolomide, and it recapitulates the patient's response quite well. So the OT naive model shrinks completely. It's really not palpable. It does come back eventually after about 120 days. And the post-relapse model almost grows straight through the same therapy. Similarly, for another pair of models, the pre-treatment and post-relapses were very different. And for this last pair of patients, I had one post-relapse model, which grew through fairly quickly, and a model derived from a patient, 1514, who only briefly had disease shrinkage, and this was much less sensitive. So in aggregate, if you line them up from least sensitive to most sensitive, they kind of recapitulate the clinical history. So I think this is sort of the stepwise approach that these models are capturing how it is that patients respond to Olaparib temozolomide as well. I won't go through the data for topotecan, which is the accepted second line therapy, but the PDX models for this disease also recapitulate whether a patient was sensitive or resistant to topotecan. So at this point, this is sort of the system that we're starting with. We have a lot of different models, so intratumoral heterogeneity is intact. There's some molecular fidelity, so they recapitulate the genomic characteristics of the patient's tumor. And probably most importantly for looking at cross-resistance, they're capturing how the patients respond to DNA damaging therapy. So if there are any questions at this point, I'm happy to run through. Otherwise, I'll go through oops, let me see. Otherwise, I'll go ahead and go through the talk.
Actually, I have a question.
There is one question.
Yeah.
Go ahead, Vito. You go start.
Okay.
Go first.
Well, so I just want to make sure I understood this. This is a critical slide, right? So, so what you're saying is that when you collect CTCs from, I wish I could. I don't think I can. No, I can't. So when you collect the serial number, let me see. If you go to slide. So you collected some CTCs from tumors prior to OT and they were sensitive.
Yes, this one right here. Is this it?
That's right. That's right. The OT naive.
Yes.
And so OT naive, and so it responded in the patient.
Mm-hmm.
It also responded as a PDX. It was derived from the tumor.
That's right.
But then the OT naive also will relapse in the mouse.
Oh, no. This is from the patient. Sorry, that's important. So the OT relapse is from the patient. So there's two models derived from the same patient. This patient at baseline before she's received OT, and that's the OT naive model. And at progression, a second set of, this one happened to be a biopsy derived, but a second model was taken from the patient. And at that point, that model was highly resistant because the patient had grown through Olaparib temozolomide. The same with 1528. So
okay.
At baseline, before patient goes on therapy, this one was CTC derived, very sensitive to Olaparib temozolomide. After the patient's progressed, the model derived at that second time point from the patient is highly resistant.
But if you take the OT naive,
mm-hmm.
in the mouse now, okay?
Yes.
So, and you treat it. So you can show you that you treat that is naive, but eventually it will relapse.
Yes.
So, okay.
That will also, that's also, so you can generate resistance that way as well. That's traditionally how resistance to therapy has been studied. But the problem with that is that doesn't always capture the same resistance mechanisms that you get clinically in patients. So
okay.
I've tried to stay away from the engineered resistance, if that makes sense.
Okay. Yes, absolutely. Yeah. Nice.
So we have two questions.
So there are a couple more questions. Yeah. Go ahead, Kavya.
Okay, so from Michael Freeman, are the tumors SC, I'm assuming that means subcutaneous, or orthotopic?
These are all subcutaneous tumors. So we're starting to make orthotopic models now, but I wasn't exactly sure what an orthotopic model of small cell would really be. And what I mean by that is at diagnosis for almost all of our patients, small cell is disseminated. And in fact, the primary tumor is often quite small, and it's the metastases that are very large. So if we're looking at the tumor that we treat in patients, it's usually in multiple different organs, including sometimes patients will have subcutaneous nodules. So it's not an entirely artificial environment for the tumor to grow in. I think the bigger artifact is growing in a mouse as opposed to with no immune system, as opposed to a human.
So a quick follow-up with that. Do you sum all of the tumor volumes together for a single patient? You're showing them essentially as single lines on these graphs, I'm guessing. Each one of these is a...
So for these graphs, these are mice.
Those the mouse.
Yeah, so
right,
these are the mice. For a given patient, you'll usually, if you're following it for a clinical trial, you usually some sentinel tumors that you follow, but you won't sum every single dot on the CAT scan. For the mice, these are caliper measurements of subcutaneous xenograft. So it's just a single tumor.
Okay,
Kavya, do you want to ask Portia's question? Yeah.
Yes, it's from Portia Thomas. She says, great talk thus far. How representative of the tumor do you think CDX and PDX are? And then have there been comparisons between patient tumor response and responses seen in CDX and PDX?
Sure. So that's essentially what this ends up being. So I think that it's always a model, and so what I've been kind of careful to do here is to try to compare at each point how the PDX or CDX model looks or responds to the patient from which that model was derived. And so what's written up here, this post-relapse stable disease or partial response, that's the patient's response to the same drugs. So with these blue models, they came from patients who had good durable partial responses to Olaparib temozolomide, and the PDX responds the same way as the patient does with a good durable response, the subcutaneous xenograft. Those arrows connect, these are separate models derived from the patients after they've relapsed, so after they're resistant to the same drug, and the models derived from them are also resistant. And then the model derived from a patient who had an intermediate response is intermediate. So I think for Olaparib, Temozolomide, Topotecan, and platinum etoposide, which are the standard drugs that we give to these patients, they capture those pretty well. If you're talking about immune checkpoint blockade, the NSG mice doesn't have an immune system. So that's sort of at the extreme end of it can't possibly recapitulate the clinical response to those, and that's a major problem. Cool.
So I just have one other quick question.
Yeah,
you mentioned that most of these tumors are clonal. You said that there's a lot of essentially background mutations. I'm just curious.
Right. The allelic fraction is high.
If that's allelic fraction. Okay, so is that consistent as well when you show in these mouse models? Are you showing the consistency of clonality, if you will, even in these ex vivo tumors, PDX and PDX?
Yeah. Yes. So the way that we calculate it, so it's based off of allelic fraction, but it ends up being cancer cell fraction to account for sequencing coverage and probably most importantly copy number. So an allelic fraction of 1/4 for genomic duplication would be essentially 100% of the cancer cells if there was for a single alteration. And this is the results that we got when we compared the PDX with the patient were the models were incredibly similar in terms of their genomic alterations, same alterations in patient and model. The cancer cell fractions that we were measuring in both the patients and the models this was done in collaboration with Roman Thomas and Julie George, who published the main whole genome sequencing effort for small cell lung cancer, and the cancer cell fractions that we were getting were very similar. What they had found was that compared to non-small cell lung cancer, the cancer cell fractions tended to be much higher for alterations in small cell. So a much more monoclonal, less polyclonal disease, if that makes sense.
Gotcha. Thank you.
Which is weird because it also has a really high mutational burden. So it doesn't, those two are kind of going in opposite directions. So this was sort of preamble to, and this was the published stuff. And then what we're starting with is, what do we do with this system? The most clinically relevant question that I found was this question of cross-resistance, since this is what ends up killing small cell lung cancer patients. So our approach is to, for approaches that we can validate, to do molecular profiles across the panel of models, 72 of them, whole genome sequencing, which is, we're in the middle of RNA sequencing, which I'll show you a decent amount of, we've gone through most of the models with bulk RNA sequencing. Mapping of enhancers, super enhancers by H3K27 acetylation, reverse phase protein array, which is in collaboration with Lauren Byers' group at MD Anderson, proteomics here, and metabolomics here with Ralph de Bourdinis. And we're at differing stages of completion for all of these. And we match these up with the functional profiles of the same set of models. So here depicted Olaparib, Temozolomide, time to progression on the y-axis versus platinum etoposide, time to progression on the x-axis for the models. And what we want to compare is the subset of models that are resistant to both first and second line therapies versus the pan-sensitive models, which tend to be derived more from chemo-naive patients. And that's how we're going to get our candidate set of features. And I'm just going to go through the first feature that we've found so far, expression feature of cross-resistance, which we are in the middle of testing now to start my lab. So the way I'm going to depict this, I'm going to again use this pair of serial models as an example. So these models were derived from the same patient before EP and after OT, and the pre-EP model was highly sensitive to platinum etoposide, to Olaparib, Temozolomide, and to Topotecan, which that patient had never seen. the post-relapse model resistant to EP, resistant to Olaparib temozolomide, and also highly resistant to topotecan, which again, patient had never seen. And so when we look at the nice thing for these three regimens, which is just very convenient experimentally, is that the mean time to progression across the entire panel between sensitive and resistant models tends to be very comparable. So I don't have to do some sort of a normalization. You can just average them. With the most sensitive models tend to have a time to progression for a therapy they're sensitive to of about 100 days. That's sort of the extreme. And so here, this is the time to progression on average, and this is averaged from 3 to 6 xenografts per model per therapy. Time to progression for platinum etoposide, Olaparib, temozolomide, and topotecan. This is for the pretreatment, 15-18-1B, very sensitive, long time to progression. Post-relapse, 18-3, very resistant to all three therapies, short time to progression. And so we haven't finished, the reason that I'm doing this, in some cases, we haven't done all three regimens across all three models, but I've done at least for 51 of these models, at least 2 regimens. And so can get something approximating an average. At some point, this entire data, the functional data set will be complete, but I don't want to wait to start analyzing it until until we finish doing all of the functional analysis. 51's enough to start with. And so there's a range of averages, but in general, you can see that for these models ranked from most sensitive to most resistant, all three regimens tend to follow with one another in terms of their average time to progression. And so if we want to look at the extreme ends here, looking at the top quintile versus the bottom 2 quintiles for the for the most resistant versus the most sensitive, and compare that to the transcript levels from RNA sequencing across untreated tumors, the strongest expression feature that I found was a strange one. It was interferon stimulated genes, which are here superimposed on the volcano plot in red, tended to be upregulated in the cross-sensitive models and relatively downregulated in the cross-resistant models. And as an overall pathway expression feature, that was the strongest one. A number of these actually rise to significance, meaning that there's a large magnitude of difference on average between these cross-sensitive and the cross-resistant models, and it's statistically significant. And I had RPPA data, so this is now at the protein level, looking at protein levels across the same series of models from Lauren Byers' group at MD Anderson. So the reverse phase protein array, instead of looking at 18,000 protein coding genes, it's looking at 208 peptides, some of them post-translational modifications, some of them the actual protein level. And the magnitude of difference and the significance was lower overall than for the RNA sequencing. But this protein, STING, came out up at the top, and it's quite, there's a very clear relationship between STING and interferon stimulated genes. And in fact, STING, although it's not itself an interferon stimulated gene, also is significantly, I think it's this one, is significantly different between the sensitive and the resistant. And so what's shown here, this is the heat map of the individual interferon stimulated genes, or ISGs, that go into the signature, and the aggregate and an aggregate signature score, which seems to work pretty well in distinguishing the sensitive and resistant cohorts. And then both the mRNA and protein level by quantitative Western blots of STING, which can regulate interferon stimulated gene expression. They both seem to be pretty strongly different between the cross-sensitive and cross-resistant cohorts. So how does this work? So STING is, I'm just going to go through very briefly the CGAS-STING pathway, and then the conundrum will become pretty apparent pretty soon. So CGAS was actually, which is upstream of STING and activates STING, was discovered here a couple of floors away from me by James Chen, and it's a cytosolic DNA sensor. So it can be activated by cytosolic DNA presented by viruses, and it's part of the cell's innate antiviral sensing mechanism, but it can also be stimulated by DNA damage when micronuclei, which contain damaged DNA, are exported into the cytoplasm and can be detected by CGAS. And sort of they mimic a viral infection. It then activates STING through production of cyclic GAMP, which then downstream of STING can activate interferon production and interferon signaling. And the interferon production and interferon signaling can, the best described mechanism of action is activation of the adaptive immune response and T cells. But the conundrum here is that in NOD-SCID gamma IL-2 knockout mice, there is no real functional adaptive immune system. So why would this be marking sensitivity to DNA damage in this particular context in a way that recapitulates, in a context that recapitulates patient responses? And it's going to have I'm just starting the lab, so it's not going to have a satisfying conclusion. We're testing this. So the two general classes of possibilities are that STING is a marker of sensitivity to, or silencing of STING is a marker of cross resistance, and the same upstream alteration affects both, or that STING is actually sensitizing small cell lung cancer to DNA damage. And if that's happening, it's in a manner that's independent of an adaptive immune response. So one first question, I'm going to sort of end on an experimental negative note. Can we use small cell lung cancer cell lines to test this? Doing all of the experiments in vivo and PDX models is expensive, time-consuming, and then there are certain mechanistic studies that you just can't really do in a PDX easily. And so to remind you, in the PDXs, the cross-sensitive models are on average have a much lower time to progression than the cross-resistant models, and they have low expression of interferon, of ISGs, and of STING. In the cell lines, they've actually been screened with a lot of similar drugs here showing topotecan. And one of the things that best distinguishes chemosensitive from resistant cell lines is whether they grow in an adherent or suspension pattern. And here it's showing that the suspension cell lines, small cell, about 2/3 of them grow in suspension, are more sensitive to topotecan than the adherent cell lines. Unfortunately, for using them to test this hypothesis, the adherent cell lines also express a lot higher. It's exactly the opposite. The adherent cell lines express more interferon-stimulated genes and express more STING. So the short answer is we probably can't test this directly in the cell lines because we have exactly the opposite relationship between drug sensitivity and gene expression. But why is kind of interesting. So small cell lung cancer tumors was found about 10 years ago. Well, it was actually found by John Minna and Adi Gazdar 30 years ago, found in the genetically engineered mouse model 10 years ago by Anton Burns, can give rise to both types of cell lines. A single tumor can give rise to an adherent or in a suspension culture, and these are quite different in terms of their expression profile. And Israel Cañadas, a couple of years ago, started to relate these two. He had figured out that if you treat a suspension line, H69, with HGF, it will sit down on the plate and become attached. And one of the main features of this transition is that it starts to produce interferon and interferon-stimulated genes. So in fact, we specifically can't test this hypothesis in the cell lines because there's only one of each tumor, and the models that we're growing are probably more similar to the admixture of both, and you get exactly the opposite expression relationship. So I'm ending on that point. I'll just sort of summarize the findings that I went through. We built 72 small cell neuroendocrine cancer models from 50 patients from a number of sources, including a lot from circulating tumor cells. The genomic alterations were retained from the patients and were stable with passaging, which is important. And probably more importantly, the PDX models recapitulate the clinical responses to first and second line therapies. The strongest marker of cross-resistance that we found expression-wise was low basal expression of interferon-stimulated genes. And what may underlie this is STING expression, and then we have to probably figure out how to test this directly in the PDX models. So I will go ahead and thank a number of people. So I have to thank where I came from at Mass General, my PI, Nick Dyson, my clinical collaborator, Anna Farrago, who just moved to Novartis, my friend Marcello, who's a fellow postdoc who came at the very end of my time there and is carrying some of this work forward in collaboration, and a strong team of technicians, a lot of models and a lot of analysis. Sarah, David, June, and Edmund, June and Edmund are still there. And then my new laboratory, which is just getting staffed. Shriyoshi, who's a postdoctoral fellow, Mitch, who's a HEMONC fellow, and Brayton, a technician. And as mentioned at the beginning, the biggest thanks is to John Minna, who brought me here, and collaborators and funding. And I think we can now really stop for questions.
This is where everybody would have a big round of applause. Thanks, Ben. Question or / comment from Michael Freeman. He says, other interpretations that there is a change in DNA damage repair. Do you want to address that?
The question is what are the other interpretations about whether or not there's a defect in DNA damage, or is the question whether there's a defect in DNA damage or DNA repair?
Less of a question, but actually, Michael, if you want to say a few words here, I just unmuted you.
Am I on? Can you guys hear me? Okay. No, I was wondering about the STING response. So it could be that the STING response is a consequence of, initially, we have a very good repair of damage, and we have small pieces of DNA coming out, say from non-homologous end joining, but we switch over perhaps to another repair system that's much more efficient. And I'm just wondering if that's why, that we see the STING response in the sensitive and not in the resistant.
So I think that, that could be, I think that the model that you're describing, I don't know if you can see my
yes, I can.
cursor or not. I believe it falls into the latter category, right?
Okay.
Like it would be so this was, when I was drawing this, I was trying to be as broad as possible and make as few assumptions as possible with the model. In the very first experiment, that's definitely possible. There's a couple, if I was to guess, I can come up with a couple of reasons why it might be a mediator and a ton of reasons why it might be a marker. A marker, by the way, for cross-resistance in small cell from a clinical and translational perspective would be incredibly useful, because if we could find these patients, not every patient following relapse is immediately cross-resistant. We give a fixed number of cycles of therapy up front, and sometimes when patients relapse, they're still sensitive. Right now, the only marker we have for whether they're going to be sensitive is how long of a chemotherapy-free interval that they had. But if we actually had a molecular marker, we could prioritize patients for going into one trial or another. It would be very useful. It'd be way more interesting biologically if it's a mediator, but that's the first question.
Well, if it's a marker, I think you're absolutely correct. It would be huge.
If it's so the best I can tell from the preclinical system that I have, where I've really tried to make sure at each point that it's capturing how the patients respond to therapy and what their tumors look like. This is a reasonable thing to test in a large tumor microarray of well-annotated, clinically well-annotated samples, where we know exactly how sensitive the patient was to DNA damaging therapy at that time point. That's a hard thing to come by. But that's sort of, I don't know what other preclinical evidence we would have to come up with to say this is a good candidate as a marker.
Okay.
But as a mediator, sure, absolutely what you're saying.
Well, while I have the voice, congratulations, absolutely fantastic research.
Oh, thank you very much. Another model for it being a marker and not a mediator is it could be that when DNA damage, that there's some additional damage that's incurred from the adaptive immune system in cells that can actually express STING. So in cells that are damaged, they are doubly damaged by then having the cGAS STING pathway activated and generating an interferon response. So it could be that the cells that initially are drug tolerant are the ones that don't generate STING, and then from among those drug tolerant cells, you have emergence of true resistance, which might be by upregulation of homologous recombination, for example.
Right, we have a question from Vito. Vito says, I noticed ZEB1 overexpressed and resistant. Is it fair to say that the resistant tumor or cells tend to be non-neuroendocrine?
So that didn't pan out. ZEB1 is a factor that can mediate the epithelial to mesenchymal transition, and I think that it's not strong, but that's not strong over the, it's barely significant and not unbelievably strong, but it's probably strong in a subset of models. If they're more mesenchymal, they're probably going to be in aggregate less neuroendocrine, but I think it's probably more specific for mesenchymal expression program, if that makes some sense, Vito. But what do you think?
Yeah, I mean, I've seen some data on the non-neuroendocrine, and ZEB1 doesn't jump at you, but I'm getting a sense, we're looking at data from buyers, from also data from the mouse model from Trudy. And it looks like there seems to be a tendency for, well, like you said, so the neuroendocrine cell lines, for example, and probably the tumors are very sensitive to, the most sensitive to chemotherapy.
Right.
And so I've been wondering whether there is this trend. I don't want to say that it's a switch, but whether there is a trend for the relapsing tumors or resistant tumors to be more in the non-neuroendocrine camp.
So it's certainly possible. I just don't exactly know how to make sense out of the cell lines versus, at least for, I don't know how to exactly translate the cell line data into the patient or the PDX data. And so when I was bringing up this example, absolutely the suspension cell lines that are more sensitive to topotecan, for example, are also more neuroendocrine than the adherent cell lines. The adherent cell lines express more ZEB1. But at least in this case, Israel probably figured out why, they also have a ton more interferon response, if that makes any sense. So it's sort of, and I think that we run the risk sometimes of conflating intertumoral and intratumoral heterogeneity, although not you don't run that risk. I think that you,
no, I mean, but I know what you're saying. Since I am here, so by the way, this was a great talk. Okay, I really enjoy the system that you have. You have a long ways to go, many years to work on this. So I'm glad you got the CIPRIT and the Texas funds, to go forward.
Yeah,
but also when we talk about DNA damage in the pre-treatment tumors, we're talking about, besides the mutations, we're also talking about DNA damage that came from cigarette smoke, right? So DNA adducts, these kind of things.
Right.
Okay.
So, so in terms of resistance to DNA damage, I guess when I'm talking about resistance to DNA, I'm talking about the resistance to DNA damaging therapy, and the specific phenomenon that I'm talking about is going from a 70% response to a 10 to 15% response to, you know, that entire class of therapies. But this tumor that arises, that's 70%, you know, that's highly sensitive to DNA damaging therapy, has already been bombarded by, you know, 100 pack years of cigarette smoking.
That's right. And so maybe the gas system is already, you know, detecting these things. Okay, so thing comes up. Okay, makes perfect sense. Great story.
My pleasure.
So as a follow-up to that, do you have any evidence of genetic alterations themselves being involved in this relapse, or are you thinking it's a non-genetic mechanism? What are your thoughts on this?
So I end up coming to the same consensus that's starting to get published, and I think just about everything that we've seen so far comparing small cell models or patient tumors pre-treatment and post-relapse is that resistance is probably not genetic. It's not going to be the same as resistance to MAP kinase therapy with, you know, a bypass track mutation or something like that. This isn't a coherent pathway. So, you know, we won't get V600E for, you know, EGF with an EGFR TKI or a MET amplification. It's going to be, and we're not seeing any recurrent alterations in those studies, either in the models or in the patient tumors. It seems more likely to be epigenetic.
And right now, your best guess is this STING pathway. That's the most common across all of these resistant phenotypes, or are you so what other areas are you investigating in terms of the potential molecular mechanisms of allowing these cells to overcome the treatment?
Sure. So so where this is eventually going to go, I guess I don't have it here, but where this is eventually going to go is we'll test individual candidates as they come up, just to get started, but especially if it looks like it would be useful either as a marker or mediator. But ultimately, we're assembling right now a list of candidates to go into a very targeted in vivo screen, because it would be a lot of work. And so like 20 to 50 candidates, and the best 3 that I have so far in terms of pathways are c-Myc. So this model, this 15-18-3, I didn't show it here, but it acquires on the way to becoming highly resistant. The 18-1B has very low levels of c-Myc expression. It acquires a focal and huge c-Myc amplification during these first 2 lines of therapy, and then this model is incredibly resistant. So it's it goes from being essentially a c-Myc non-expressor to the highest expressor in the panel, and the c-Myc amplification alters the expression profile of this model so much it doesn't even cluster on a Tisney plot anymore. I freaked out the first time I saw it. I thought I'd messed up the name of I thought I'd messed up the patient, but the DNA fingerprinting, it matched up, and it just radically alters it. I think that happens in a subpopulation of patients. How it is that c-Myc amplification would confer cross resistance mechanistically, the first thing we're going to test is whether that happens, whether it does, but then how it would do that, I think, will be interesting. And then the third is, as Vito was pointing at, ZEB1 is an example, but expression of the mesenchymal transcription program, I think, accounts for a significant proportion of cross resistance. It's not going to be the same mechanism for every patient, is the thing.
Yeah, so that's a very interesting question about the specificity for each patient in terms of the pathway that they're taking and what ways that they have access to these pathways, whether they're restricted in some way, whether they're already essentially set up to follow a particular resistance path, or whether they just choose one randomly.
Oh, that'll be, I mean, if we're lucky enough to find more than one that actually occurs in patients and that we can verify, validate in a pre-treatment model, then, like, for instance, for this pair of models, if we were lucky enough to find 2, then like doing the test of, do they both work in this model? Does only one, is this model preordained, as you said, to only become cross-resistant by acquisition of a c-Myc amplification? That would be such a fun experiment to get to do. We've got a long way to go before we get to do that, but it would be fun.
Very exciting.
We have one last question from Michaela Elder. Are there similarities in the roughly 30% that don't respond to initial treatment and those that become resistant to the second treatment? And then is there a common mechanism between those populations?
That's an awesome question, and I think it's really understudied within small cell lung cancer, the primary refractory platinum etoposide cases, and the problem is there just aren't that many clinical samples from them. So I don't think any of I have one model from a patient who actually grew through platinum etoposide. A lot of the time, and this actually has to do with the pathologic annotation of these tumors. A lot of the time when it's primary refractory, we go back and we end up finding out it actually wasn't small cell. It was a large cell neuroendocrine tumor, or a neuroendocrine tumor with an adenocarcinoma with neuroendocrine features, which are much less sensitive to first-line therapy. Only 30% or so would respond to platinum etoposide. So I don't think that we have a large enough collection, at least not that I've seen or that have been published, to be able to answer that. And so therefore, I don't know if it's going to be the same de novo or acquired resistance mechanisms. I would guess not. Usually it's not. Okay. So should I stop? I'm going to stop sharing.
Yeah.
Yeah, that's great. So thank you so much for an excellent presentation and answering all these questions. The next session, we're going to go to a different Zoom link for the student-led portion of the discussion and further systems level thinking. I put the link in the chat. Unfortunately, we can't just stay on this link. We're going to have to switch over to a different link. And hopefully everyone that is interested can just hop back on. And all students and postdocs are welcome to attend. So if you can,
all right. So
copy that link and we'll restart momentarily.
So I should go now, right?
I think so.
Okay.
All right. See everyone over there. Thank you.
Thank you.